Okay, so we've talked about smooth muscle, we've talked about cardiac muscle, and now kind of for the main event, we'll go ahead and talk about skeletal muscle. And this is by far the most famous, right? This is what moves all the bones in your body, allows you to walk, allows you to stand up. Then you should already know that these are voluntary, right? And striated. So we can talk about some of the functions of skeletal muscle. Some of them may surprise you, may not. Of course, the first one is body movement. And the next one will be actually maintaining body posture. Right, because if you don't have muscle strength, you can't even stand up straight or hold body posture, so you'll just fall. So it's important to know that just standing straight uses your muscles. And you utilize that in physical examinations all the time when you just see someone just sitting there, and whether or not they can sit there and maintain their body posture will tell you actually a lot about what's going on. And the last one is actually to maintain body temperature. And you know that too, of course, on a cold night when you shiver. You probably never knew why you shivered, at least I didn't when I was a kid. But of course you get cold and you shiver and you're generating all this heat, and it helps you stay warm. So that's also another important function of skeletal muscle. Now, two important concepts I want to introduce here. I'll just change the color because it's nothing related to the function really. Is origins. That blue. Origins and insertions, and you'll hear these words a lot. So I think it's important to understand what they mean. Even though I've heard different definitions and different texts, and even when I was researching it, it seems to vary a little bit. But for the most part, an origin of a muscle. So we're talking about the origin and insertion of muscles. So where they come from and where they go. So for example, a muscle can start on the humerus and end up on the radial bone. That's kind of what we're talking about here. So the origin is normally more proximal, and it's normally more of an anchor point. Oops. Normally more of an anchor point for the muscle, as opposed to the insertion, which is normally more distal. And we're talking to the the median of the body, so proximal, closer to the the sternum, and then when we're talking about distal, like closer to the lateral part of the body. So this is normally more distal, and normally is more involved with moving the muscle it's attached to. It's important to understand these 2 points. You hear it a lot when you look up muscles that say origin and insertion, and just so you kind of are familiar with those terms, I wanted to go over them with you. So now let's go into what exactly a muscle cell looks like. So let me clear this. Important concept actually I want to talk about before we move on to a diagram of a muscle cell is that a muscle cell is sometimes interchangeably referred to as a muscle fiber. So those are two of the same things, just in case you get confused, because normally when I think fiber, I don't think about a cell. I think of an actual piece of fiber. So these are actually two of the same, muscle cell and a muscle fiber, which will kind of make sense in a little bit when you see the structure of a muscle cell, because it's pretty much just long myofilaments, and it kind of makes up like a fiber type of design. So let's go ahead and move on to a diagram of a muscle cell. So this is a diagram of a typical muscle cell, obviously very basic, but I just wanted to get the general idea of what's going on here. And let's label some of the stuff. So one histological feature of muscle cells that are completely different than skeletal, than cardiac muscle cells and smooth muscle cells is that they are multinucleated. And you could probably guess that these are them right here. This is a nucleus. Also be aware that these nucleuses are peripheral. And that's kind of important. So when you look at a histology slide, you can see these kind of squashed up against the membrane here, these nucleuses, and they're multinucleated. They're not just two normally. Normally it goes on, and these nucleuses keep adding up on the side. So there's normally a ton of nucleuses. Now these nucleuses sit right underneath the membrane here, and this is sometimes referred to as you'll see it written as just to confuse you more, as a sarcolemma. And that normally just means thick membrane, because they didn't really call it a cell membrane before, but now they know it's a cell membrane. So, but sometimes you still see it referred to as sarcolemma. And also the endoplasmic reticulum, sometimes referred to as a sarcoplasmic reticulum. So that's just to confuse you, but really the same thing as a cell membrane and an endoplasmic reticulum. Now these green horrible lines that I drew here are the muscle, what are myofilaments. Let me label that. These are myofilaments. And they span the whole entire range of the cell. And these are the contractile elements of the cell. And you can imagine when it contracts, it's attached to each end because they span the whole, all the way through the muscle cell, and they bring the whole cell together. And actually in the next slide here, we'll take a chunk. And these right here are my make-believe striations, just because you got to remember these muscle fibers are striated. And in the next slide, what we'll do is we'll take a chunk of this myofilament, and we'll break it down even further, just because it's important to know the structural units in these myofilaments. Just wanted to give you a reference of what we're going to be doing next. And okay, so let's take a look at that. Okay, so now we have this myofilament here. So let me label this myofilament, right? And we broke it down into this contractile unit, and they refer to this whole thing really as a sarcomere. And I just want to make a distinction real quick between the kind of units they use in muscle cells. So you have myofilaments, right? Myofilaments, and you have multiples of myofilament or multiple myofilaments in a muscle cell, or as we said is the same thing as a muscle fiber. Okay, so you have multiple muscle fibers or muscle cells, right? Because we said those are pretty much the same thing, and where they actually are the same thing, in something called, they refer to as a fascicle. Right, so we have multiple muscle fibers that make up this fascicle and multiple myofilaments to make up a muscle cell. So this right here is what we're referring to up here. And then you can further break this down, and this is that famous striations we always refer to are these sarcomeres that I have drawn here. Now let's go over the concept of a sarcomere, because it's important to understand the terminology with this as well. So you may remember from previous class or anything, thick and thin filaments that make up this sarcomere. And so we have actin, which is referred to as a thin filament, and then myosin, which is a thick filament. I try to represent those up here, but of course it's not the best, but this thick band right here will represent myosin, and this thin filament right here will represent actin. It's important to understand the terminology they use when talking about sarcomeres. So let me erase some of this stuff just so we can have some room to write. So they've named different zones of the sarcomere with different terminology. So we'll go over that real quick. So remember, this is thick filament, thin filament here. So this part right here that just includes the end of the sarcomere, and right where the thick filament starts is called the I band. And this is just thin filaments, right, as we labeled. Just thin filaments, just thin. And then the part that includes just thick filaments, right, is called the H band or H zone. And I just remember it because it's heavy, H, it's thick, thick and heavy. So this is just a thick filaments. Now another important one to know is these zigzag lines as I represented them here. This is called a Z disc. There you go, that's how they refer to those. And then another one that's important will include everything in the range of the thick filament, including whatever thin filament is overlapping. So that's important to understand. So it includes everything in the thick filament and then whatever thin filament overlaps, as opposed to the H zone, which is just thick. And then once the thin filament comes in, it doesn't include that. This is called the A band. Now, when you look at muscle cell histology, which we will do in a second, you see Z discs and A bands is pretty much what you can see as striations. And it's important to understand these when we talk about contraction here in a second. And there's also something called, let me draw it in white here, an M line, which is directly right in the middle of the thick filament, M line. So when a muscle contracts, and this is important, muscle contracts, the part that contracts, they call the His. That is the part that actually, well, not contracts, but that's the part that gets shorter. So it's important to understand that. So the H zone is getting shorter, which makes sense because when this thing contracts, these thick and thin filaments will start going towards each other. So this during contraction, this is going to go this way. This is going to go this way. So the H zone, the part that is just thick filaments, is going to get shorter. So that makes sense, right? And that'll account for our H here. And then the I band as well is going to get shorter because this simultaneously is going this way, like that. So it makes sense that this is coming together as well. So our I band is going to get shorter. And then the Z disc, which is our whole thing from this side to this side, will get shorter because the inside is contracting. So this Z disc coming in, this one is coming in. So the Z disc is going to get shorter. So that makes sense. Now, the one that kind of got me for a bit was actually the A band does not change. So that's important. So A band does not change. And it's important. At first, I'm like, why doesn't it change? It doesn't make any sense. Well, the A band will always consist of the length of the thick filament in the middle here. So no matter what the thin filament is doing. So even though the thin filament is coming in, it doesn't really make a difference. It's going to come in here. The thick filament is still going to be the same length. So the A band technically just consists of the entire thick filament, including whatever thin filament is there. It doesn't matter if it's all the way contracted or all the way stretched out. It's always going to be the same. So the I band always stays the same. And some people remember that as A for always. Stays the same. Whatever helps you memorize it. Okay, so let's now move into how, or the process of how the muscle contracts. Okay, so now let's talk about how a muscle cell is told to contract. So let's do, let's title this muscle cell contraction. And I drew a stereotypical neuron axon right here. And then this is what would be our muscle cell. So let me just label this muscle cell. And this will be our neuron right here. Now, well, first thing will happen is an action potential will come through here, right? And this will get depolarized. And like a classic neuron, calcium will shoot through here. And what you'll have here waiting on the other side is some presynaptic vesicles. And when this calcium comes in, it'll cause these vesicles or assist in helping them dock and fuse to this membrane, and then shoot out its vesicle contents, which in the case of a skeletal muscle, what really activates this cell to go off, let me draw it in blue here, here's a receptor on the muscle cell, is acetylcholine, which is pretty famous for doing this. So acetylcholine will shoot out here once this vesicle fuses, just like that. That's acetylcholine. It'll bind its receptor right here, and this will start the process. So that's how it first initiates, and then what it'll do once it binds here is it'll actually cause this membrane to depolarize. And this depolarization of this, what they call this is a motor end plate. It's just pretty much the portion of membrane that is coupled to this neuronal axon. And this depolarization will travel down, and it'll travel down both ways here, all along the membrane. And then it'll actually travel to something called a T-tubule, which is almost just like an invagination of the muscle cell membrane. So I'm just going to encompass this for no reason, but this will be like a T-tubule right here. And then the action potential will continue cruising down all the way down this T-tubule, and it'll hit a, where is that? It'll hit a receptor, and this receptor is called a dihydropyridine receptor. We can actually just pretend that's it right there. So it'll hit this right here, and then it'll cause a conformational change in this, which will cause a conformational change in another receptor. Let me draw that. We can do it in green. Right here, that's connected to, this will be the sarcoplasmic reticulum. And we said earlier, this is the same thing as its fancy name for endoplasmic reticulum that we're all used to. And then this receptor is actually really important right here. So let me just mark it out here. And it's called a ranodine receptor. And then, so this is all triggered by the depolarization, comes down, hits this dihydropyridine receptor, which is mechanically coupled. So that's important. So it's mechanically coupled. So let's mechanically. So yeah, it's mechanically coupled. And that's important because it does not rely, write this down, it does not rely on extracellular calcium in order to initiate this action potential. So that's important because we talked about cardiac muscle, which relies on extracellular calcium. Skeletal muscle does not rely on extracellular calcium, just for this reason that it's mechanically coupled to this ranodine receptor. And then once it hits this ranodine receptor, calcium on the inside here, which is stored in this sarcoplasmic reticulum, gets released. So that's released out into the muscle cell, and then from there it will cause contraction. So this whole thing is gonna start building up with calcium, and that's really important for contraction, which we could probably talk about in a future video. But that is what pretty much directly acts on the proteins that hang out on the actin thin filaments. Well, yeah, let's actually talk about exactly where this goes. So the calcium, it will bind to something called troponin. Hope I'm spelling that right. Troponin C, and this is located on the actin filaments. And actually, it binds to this troponin C, and it causes a conformational change in this troponin C, and that will cause tropomyosin to move. Sorry, it's getting a little sloppy here, but it'll cause tropomyosin to move. And the important part about that is tropomyosin is actually blocking the myosin binding site on actin. So once calcium binds to troponin, which induces a conformational change in tropomyosin, then it will move out of the binding site for myosin, allowing the myosin head to bind to actin. And then let me erase this here just so I can have a little bit more room. And then once myosin can bind into the myosin binding groove on actin, then the next thing what will happen is, so now it's myosin binding, and it'll then cause a power stroke, which will actually just literally be a stroke. The myosin head will actually pull the actin back, and then it'll release ADP and a phosphate. So that pretty much starts this whole cycle of power stroking and contracting of these muscle filaments. So now we can go ahead and take a look at some actual tissue, which is much funner. Yay, so let's move into that. So I wanted to show you the zoomed out picture of the histology tissue that we'll be working with. And this is it, quite beautiful, and we'll go ahead and zoom up on a portion of this now. Okay, so this is our beautiful section here, and I'll go ahead and tag it up. And actually in the corner here you can actually see what part we're looking at. If you just look at this box here, there we go, and there's a little red box in there, and that's the little section that we're looking at right now. Now each one of these strands going up and down, I can mark one out here and here, is a muscle fiber or a muscle cell. So this entire thing is one muscle fiber or muscle cell, and it's kind of obvious in this section that these are this is multinucleated, right? And this is even though it looks like it's in the middle of the cell, it's not. It's actually sitting on the membrane on top. So this is just underneath the membrane, which we called a sarcolemma. And this is a nucleus right here, a nucleus here, nucleus here, nucleus here. As I said, there could be multiple, so you just got to remember it's multinucleated. Now the key feature histologically of skeletal muscle are these striations, and you can see them, these lines. See if I can mark one here. Are these lines that kind of go across this cell. One right there, 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 there. And you can see them all throughout these myofibers or muscle fibers or muscle cells. And they're pretty well arranged there. And you can actually see dark and light, dark and light. I like to think this dark is the A band where they where the thin and thick filaments overlap. And they're both included, that's why it's dark. And then you have that I band, which is a little bit lighter, but it's much better seen on electron microscopy than it is on light microscopy. And these, it looks like just one, but there's actually a ton of myofibrils running up and down the cell, just like that, all striated. And that's pretty much what you're looking at here. So we'll do my fake striations like before, just like that. So just like had we diagrammed in the previous slide. So they run across, and they're beautifully organized, and they're quite obvious in these sections, and that's how you can really tell that these are skeletal muscles along with the peripheral nucleuses. And it wouldn't be a good video if I didn't include one clinical correlate. So I'm going to talk about Lou Gehrig's disease. And this is a disease that attacks neurons. That control skeletal muscle. And because muscle is one of those things that if you don't use it, you lose it. So skeletal muscle, once you start kind of activating it so much, it starts to atrophy. And the muscle cells start to die. Then once these muscles atrophy, then you start losing control of swallowing and speaking and eventually breathing, and that's why Lou Gehrig's is a pretty fatal disease. And that marks the end of this skeletal muscle video.